ADAMTS17 is a secreted metalloprotease that functions in the extracellular matrix of various tissues. Mutations in ADAMTS17 can cause Wheal-Marcasani syndrome, a musculoskeletal disorder that presents with short stature, brachydactyly, and eye abnormalities. We identified 2 splice variants of ADAMTS17 that are generated by alternative splicing of the spacer domain, a component of the protease's ancillary domain. Compared to wild-type ADAMTS17, 17A contains a longer spacer domain, and 17B contains a shorter spacer domain. Real-time PCR analysis shows that these isoforms are differentially expressed in different tissues and cell lines. To determine the functional consequences of the splice variants, we analyzed the conditioned medium and cell lysates obtained from 17, 17A, and 17B expressing cells. We found robust expression of 17B in the cell lysate, but no expression in the medium, suggesting either lack of secretion or cell surface retention. In contrast, 17A was present in both the conditioned medium and cell lysate, but compared to wild-type ADAMTS17, autocatalytic processing was altered. We immunostained 17, 17A, and 17B expressing cells and found that all of the splice variants were localized to the surface of the cell. We also co-cultured 17 and 17A expressing cells with human dermal fibroblasts to assess their function in the matrix. We found that both isoforms co-localized with fibrillin-1 microfibrils, but there was no effect on the quantity of microfibrils, suggesting that neither isoform functions in the proteolysis of fibrillin-1. Because no substrates of ADAMTS17 are currently known, we cannot determine the effects these splice variants may have on the catalytic activity of ADAMTS17. Nonetheless, their differential expression in tissues and cell lines and extracellular localization suggests that the existence of these isoforms is physiologically relevant. Future studies will be directed at understanding the in vivo relevance of these ADAMTS17 splice variants.